10 different tasks that are looking into 10 different technologies to be developed. I will show you today something about automation and miniaturization, especially an automated preparation unit, sample preparation unit, and solubility and reactivity assay on a chip. Large chip, but it's chip-like. Sample measurements, mass size, and chemical composition of nanoparticles. In that case, new sample introduction systems into single particle ICP-MS techniques, and data analysis and software systems that are looking into multi-element analysis on single nanoparticles based on time of flight spec. I will also give you an example on environmental samples that actually have relatively high complexity and can be resolved with these new technologies. Starting with an automated preparation of nanoparticle samples using a newly developed autosampler and sample manipulator that has been developed based on a survey on requirements, what actually users and companies that do routine testing would like to see in terms of an automated sample preparation and introduction system, also to reduce variability between samples and easing the whole measurement process. There's one example here. This is the technology. This is the sampling robot that has been produced. It can draw samples, but it also can dilute. It can mix samples and can vortex samples. It can sonicate samples online and do a filtration and then injection into various types of instruments coming like field flow fractionation, DLS, and others. I had two application examples, but due to time restrictions, I will give you only one of those two. We have run nanoparticle suspensions in a dilution series that was really successful in terms of reproducibility and variability between the dilutions. So the system can automatically dilute samples to a preset value, but it can also actually take nanoparticle powders and bring them from powder to a stable suspension fully automatically. And this has been shown on the OECD standard material titanium dioxide, the NM104 coated titanium dioxide nanoparticle used in sunscreens. The example here is using this material, put 40 milligrams into a vial, adding a dispersion medium and dilute that, then actually going on further with mixing, so drawing and dispensing, so mixing up the nanoparticles in the dispersing medium. Further on, then actually put it into a sonication device next to the autosampler that has been developed specially for this device. It's a more or less a wild tweeter type. And by supplying quite a high amount of ultrasonic power, you can easily disperse this powder sample into dispersion medium, and the results in the end are relatively well reproducible between different sets. And the DLS measurement comes up with 190 nanometers almost, and the field flow fractionation measurement comes up with 60. The difference between these two are based on the coming from the different measurement techniques. Usually the results that you get from field flow fractionation with light scattering are smaller than those from the DLS, because the DLS is much more weighted towards the larger part. So the conclusion from that is we have an in-house validated SOP in terms of repeatability, intermediate precision, overall measurement, and certainty. The technology readiness level is 6, so the demonstrator is running in a relevant environment. And we are still looking into an interlaboratory comparison for a full validation. Information are available from Postnova in Munich. Please contact them if you're interested in that device. The next one actually is a reactivity assay on a chip. So we are looking into the reactivity of nanoparticles via surface reactions. And this has been actually miniaturized and put on a well-plate reader type system. And this is how it looks like. It's a polymer-based mixing and readout system. It has 24 assays in parallel in possible, and you can see actually how the idea actually works here. So you're putting the nanoparticles and the reactant into two wells. This is automatically dispensed. And then actually you are bringing them through mixing into a mixing chamber. From this mixing chamber, and the excess actually is flowing over into a waste chamber. And then actually the reaction can take place in this chamber for a given amount of time. And we can see here actually the testing of silver nanoparticles and methylene blue. So the silver nanoparticles reduce the methylene blue to methylene blue that is non-absorbing the light. And this is actually the reaction kinetics over time in seconds here that can be nicely monitored with this device. And as I said, 24 wells in parallel. And also the measurement is made actually in the device itself. And the trick that has been applied here to assure an optical path length for the measurement in every single well, and this should, as we know, always be the same for each well to gain actually precise results. There's an approach that we are actually putting the readout probe into the well. So, and this actually guarantees from the bottom to the readout constant optical path length. And this is held constant for each single well. And this is how it looks like in practice. So you have 24 laser diodes that are illuminating our readout wells. And so you can actually read out with high time resolution and in high parallelity 24 different samples. So the chip instrument has been developed, has 24 parallel units. It's an on-chip, in-plate, automated liquid mixing, incubation, and absorbance readout. So it eases up the measurement of surface reactivity of nanoparticles a lot. We are at a high TRL level, so the system prototype has been demonstrated in the relevant environment, and we are about to run the validation tests and interlaboratory tests within the next months. Now we are coming to the section of single particle analysis and laser ablation single particle analysis based on ICP-MS techniques. The partners from Zurich, from the ETH Zurich, have developed a new sample introduction system that is distinctively different from the normal pneumatic nebulization and spray chambers, and also the desolvation systems that are currently used in ICP-MS and also single particle ICP-MS. Here's a single droplet injector. So it injects single droplets at high frequency directly into the plasma. These droplets have a size of 25 micrometers, are really highly reproducible in terms of size, and also in terms of mass and amount of liquid carried into the plasma. The frequency is at 100 hertz, so you're measuring 100 droplets per second. That, of course, with this small droplet size means that you're really having a very low sample throughput, so you can also measure extremely small sample volumes. But also you're achieving 100% transfer efficiency from your sample inject into the plasma because every droplet reaches the plasma directly. You can actually use an internal standard, a dissolved standard that is the liquid in the droplet, and each droplet carries a single particle. And this single particle in each droplet is then actually differentiated from the standard and can be nicely measured. Because of the fact that we have very low matrix loads into the plasma, we are also achieving quite good detection limits. The examples here for a gold nanoparticle go down to 3 nanometers on a sector field instrument, and that's quite some improvement towards other techniques. Of course, the sample consumption is extremely low, and also at the same time we can reach, because we are dispensing single droplets, we are able to also cope with higher nanoparticle concentrations than normal in the sample. So we're going up to about a million particles per milliliter. So also dilution requirements are much less, and the working range is higher than have this from normal injection systems. Another approach and another idea was actually that we wanted to have an option to directly measure nanoparticles on, for example, on substrates, for example, soils, without needing actually to extract those nanoparticles in a liquid extraction step, what is the current technology that we have. So we are extracting the particles with a dispersant using ultrasonic power, hope that we actually release the particles from the soil matrix, and then measuring those released particles in single particle ICP-MS. The efficiency and the recoveries of this technique is usually around 20 to 30%. So we are not capturing usually all the particles that are in the soil, because the adhesion between particles and soil is relatively high and cannot be always broken up by those liquid techniques. So the innovation was to use a laser ablation system to levitate particles from the surface of the soil. So the classical high energy laser that simply evaporates the stuff. It is a tuned laser that comes with an energy that is enough actually to levitate, to release the particles from the soil and bring them into the ICP-MS, but not destroying the particles. That has been tested on different matrices like filter paper and polymers and soils. And we are using a T-piece device to be able, in the same setup, to be able to calibrate and measure consecutively. So always being able to calibrate our instrument between runs where we ablate particles or levitate particles from the sample. We see actually that there's an impact of the matrix on this, on the calibration curve. So we would require a matrix matching for our calibration. What is actually also possible by running the ablation of soil and the calibration in parallel. Results are quite good actually when we are looking into 60 nanometer particles that are attached on our filtration membrane. We see actually that we can actually levitate the particles and measure them, as well 60 and 100 nanometer particles. The mass recovery from that actually is 40%. And when we are going to soil, to real soil, actually we are recovering even up to 85% of the particles that have been brought onto the soil. We also see that we have some fragmentation, so the laser seems to fragment some of the particles, and they actually appear then at lower sizes and lower masses, of course. Seems that the fragmentation is usually around a tenth of the original particle in terms of mass, and this is something that you, of course, can tune with the laser power that you actually apply. This is a technology that is higher efficient than the normal extraction of particles with liquids. It is very much faster than just using electron microscopy. It gives us a direct access to the nanoparticles without any further or prior manipulation of the sample. And we also see a very low alterations of the particle size, apart from some breakup effects that are obviously visible and still needs to be tackled. We can use similar calibration procedures for aqueous single particle ICP-MS with matrix matching, like simultaneous ablating of non-spiral soil, what actually covers then the matrix effects on the analysis. The recoveries still depend on the choice of the substrate. Reason is still not 100% known why this is the case. It has, of course, something to do with the binding energy between the particle and the surface of the matrix, but this actually still has to be explored. That's the reason why it's an innovation and not a standardization technique. Coming to the probably most recent developments in terms of single particle ICP-MS, and probably also the far-reaching developments with using time-of-flight mass spectrometry in single particle ICP-MS to enable the spectral analysis, the multi-element analysis of single particles in the femtogram range. TOFWERK produces this instrument, the ICP TOF-MS, in Switzerland, and they have, within our project, developed several methodologies, but also improved their software environment to ease up data treatment and also identification of single particles and events, and being able to actually do multi-element fingerprinting on those particles to say actually are these engineered or are these natural or incidental particles based on their elemental ratios, because you can simply measure almost every element of the periodic table and see if this is actually existing in your particle. Just think about what happens when you're measuring for 1 minute and you get actually 33,000 data points per second from this instrument because of the readout speed, 33,000 single measurements per second, and this over 1 minute, and this over the whole spectrum, elemental spectrum, and this with a high resolution. Then actually you will see that you're retrieving roughly, if you would measure everything, you would retrieve per measurement terabyte sizes of files with millions of information in terms of elemental distribution and particles. This has been actually optimized and further developed to ease up the process, and we have a new sample sequence management system where actually you see actually directly your time, your elapsed time, the status of your sequence, your measurement log. And if you have run actually your sequence, you go into an environment that actually helps you to automatically your samples. So single peaks in the whole system, single peaks are identified, are compared with the elemental table. And then actually single particles are identified. There's a thresholding in terms of distinguishing from the dissolved background, and split event correction if you have actually had an event that was split across several measurements. And finally, then a conversion based on calibration, a conversion from counts to mass, the wrong way around, from counts to mass, and of course also a size determination if you have an idea about the composition and the density and the shape of the particles. You can offline reprocess all your data, so you can always go back to your original raw data file if you would like to look up new elements, if they're existing in your sample or not. This is all stored in the background, and then you get actually a PDF report on calibration, limit of detections, signal intensity and mass-based histograms of your particles, and of course calculation of particle number concentration. This is all also converted into a CSV file that you can read out with several data management software, and you will have a timestamp for each single event, each single particle that arrived in the plasma during your measurement time. That actually makes it possible to see actually what kind of elemental distribution is available for each particle, and that actually gives you the opportunity to characterize and also identify the particle as from which source it might actually come. There's also a development in the laser ablation application of this instrument, and that actually is able to plot these kind of surface maps on the fly during the measurement for different elements that have been selected, and it gives you actually all relevant laser parameters and so on, and eases up this kind of spatial analysis in a quite nice way. Just imagine that you're measuring actually whole elemental spectrum within milliseconds, and that means that speeds up the laser ablation readout extremely much. So your measurement times are reduced quite dramatically, or you can increase your resolution. At the University of Vienna, we actually have seen, or we were aware about the fact that when you're measuring always, all the time, the whole spectrum, that interferences become as important as they have been in the early times of ICP-MS development, because the use of reaction cells doesn't always help in that case, and sometimes the resolution, although it is quite high, the resolution of the instrument doesn't allow you to separate isobaric or polyatomic interferences. So we are looking into techniques to identify possible interferences and using calculation algorithms and peak detection algorithms to actually reduce the risk of picking up an interference instead of your target element. One example in the end, this actually has been done in collaboration with the criminal investigators in Germany. Just something about gunshot residues. When you are investigating gunshot residues on clothing or on tissue or whatsoever on your skin, you're taking a sample from it, bring it to the electron microscope, and looking for single small particles in the micron range that contain all the three elements, barium, antimony, and lead. We have actually taken several different gunshots from different manufacturers, from different weapons, in total, I think 12 or so. We analyzed them in TOF-MS, where we can actually measure thousands of particles within a minute. Try to do this in an SEM. And we also have access easily to particles that are way smaller than 1 micron. And we are also measuring not only these three elements in full quantitative mode, we are also measuring many, many other elements that could actually be part of different origins of different ammunition and different powder mixtures in those guns. So we would be able, even in some cases, it looks like able at one point to say, oh, this is coming from this type of ammunition, this is coming from this type of ammunition. I think this will be a really advantage and a really step forward in this application, speeding up analysis and even making it more specific than it has been done before. That was a really interesting part. We learned a lot also about criminal technology investigations and what people can do nowadays. That's impressive. But we think we have added something to that toolbox from Acenano. That was from my side, giving you an overview about some things that we have developed. You have seen that actually there's TOFWork, there's PostNova, there's CSEM. Contact those companies if you're interested in these developments, or contact us if we should actually link you up to our partners. Thank you very much for listening. I would then conclude my presentation with that and give over to Aoife, and we will collect questions just after Aoife's talk.
Okay, can you folks hear me? Or Christopher, can you set me up?
Yes. Can you please share your presentation?
Okay, do I do the region share?